Today, I'm going to be discussing positive repressible regulation in prokaryotes. Prokaryotes regulate their genes through transcription, which occurs at a segment of DNA called the operon. The promoter region is where RNA polymerase binds to start transcription. The operator region is a regulatory element which controls transcription. The regulatory gene encodes a protein that regulates gene expression of this operon. It's a regulatory molecule or protein. Activator proteins mediate the process of positive control of transcription, and effector molecules will bind to a regulatory protein and affect their activity by changing its shape. An activator gene is transcribed into activator mRNA, which is then translated into an activator protein. The structural genes are transcribed and translated to make enzymes that code for proteins and effector molecules. In this case or model, an inducer is produced. This inducer can inhibit transcription and gene expression by binding to the activator protein, conforming it into a new shape that is unable to bind to the operator. In positive control, an activator binds to DNA to stimulate the transcription of structural genes. Repressible control means that transcription is usually on but is turned off when a small molecule binds to the regulatory protein. Overall, positive repressible regulation means that the activator is usually bound to the DNA, allowing for transcription to occur. But when a small molecule is present, it can no longer bind to the DNA, causing transcription to stop. Small effector molecule or inducer is absent in this case, and the activator protein can bind to the DNA and signal to the DNA polymerase to begin transcription, expressing the gene. When the small effector molecule or inducer is present, the activator protein is inhibited from binding to the DNA, leaving the DNA polymerase unable to bind to the promoter to start transcription, resulting in the gene not being expressed. Here's a quick guide of how my analogy works. The activator protein is represented by an alarm signal. The small effector molecule represents a dead alarm battery. The activator binding site is the action of getting geared up. The RNA polymerase is represented by a fire truck. The structural genes are represented by houses that are on fire, and transcription of structural genes is the action of driving to the houses on fire. In normal conditions, the small effector molecule is not present, and transcription of the structural genes will occur, meaning that there is no dead alarm batteries, so the alarm or activator protein will bind to the operator, which represents the firefighters getting geared up. This binding signals for RNA polymerase or the fire truck to begin transcription, or driving to the houses that are on fire, to get the gene product of putting the house fires out, which represents gene expression. When a small effector protein is present, it will bind to the activator protein and prevent it from binding to the operator. When there's a dead alarm battery present, the alarm or activator protein cannot signal to get geared up, and without getting ready, the fire truck will not be able to drive to the houses on fire, resulting in the firefighters being inhibited from putting out the houses on fire.